Hi, my name is Elsie Carrillo, and I'm a fifth-year PhD candidate in ecology and evolutionary biology here in the Meta Lab at the University of California at Santa Cruz. And I study the mechanisms that facilitate a semi-aquatic existence using garter snakes as a model. For garter snakes, being able to move between land and water is an important part of their natural history, since they enter the water to forage for tadpoles, frogs, and fish, as well as to escape from predators like hawks. They also, as ectotherms or behavioral conformers, enter the water for behavioral cooling and to facilitate ecdysis or shedding. So we recently discovered that when garter snakes enter the water, as an alternate strategy for a breath hold, they can prolong their time underwater by rebreathing air from an air bubble that forms over the nose or the nare, and these are called narial bubbles. This is similar to other semi-aquatic organisms that use air bubbles to prolong their time underwater, such as reed canary grass, that use these air bubbles to prevent drowning during the rainy season, and diving beetles and diving bell spiders that use air bubbles that form over their body to hunt underwater, as well as anolis lizards that use air bubbles that form over their head or their nose to escape from predators. We observed the use of air bubbles in 3 species of garter snakes: wild valley garter snakes, as well as our captive-bred checkered and Lake Chapala garters. Interestingly, the use of narial bubbles varies with degree of aquatic dependence, since our more terrestrial species, the checkered garter snakes, use them more often than the more aquatic species, the Lake Chapala garters. We couldn't just observe these narial bubbles being used, we had to test to see if garters were actually rebreathing from these reservoirs. So we probed the bubble with an oxygen sensor to measure oxygen consumption over time. And we determined that since the amount of oxygen was decreasing rather than increasing, garter snakes were indeed rebreathing air from these bubbles on the nare. As a physiologist, I needed more evidence, however, to show how this was happening. And knowing when we as humans, we enter the water on a breath hold, as a reflex, our heart slows to better preserve our breath, I used a fetal Doppler to measure heart rate while snakes were underwater. And what we found was that when garter snakes enter the water and no narial bubble was formed, heart rates were slower to better preserve the breath. And when garter snakes enter the water and a narial bubble did form, heart rates were faster to facilitate rebreathing and active behavior. So in summary, narial bubbles act as a form of natural scuba gear for garter snakes, since they are able to prolong their time underwater by rebreathing in order to perform behaviors that are vital to their survival. Future work includes looking at how water temperature based on climate change forecasted conditions influences narial bubble usage, given that warmer water stores oxygen in lesser concentrations than colder water. The work has potential to be impactful in that it will not only provide insight into the mechanisms that facilitate a semi-aquatic existence, but also allow a vertebrate group that's often overlooked due to ophidiophobia or fear of snakes to be appreciated and ultimately conserved. And I would like to thank the ARCS Foundation for allowing this work to be possible and for providing opportunities not only for myself to do exciting science, but for providing opportunities for undergraduate mentees to develop their scientific identities through research opportunities. So thank you so much again.